So I move to the next one. Dr. Anupha Prachumwat. He is a senior researcher at Thailand National Center for Genetic Engineering and Biotechnology. He received his doctorate in genetics from University of Chicago, USA. He is interested in utilizing genomic sequence to better understand interaction between shrimp, pathogens, and microbiomes. He analyzed genome and transcriptome of several shrimp pathogens for developing detection methods and strategy to control infection and reduce their virulence. He and his colleague investigate microorganisms and pathobiomes of shrimp aquaculture system for shrimp health and pathogen discovery of current and potential future emerging shrimp disease. So Dr. Anupha will give us a presentation entitled Microbiome Signatures from Stocking to Emergency Harvest in Thailand, Penaeus vannamei pond experience yellowhead virus outbreak. So Dr. Anupha, the floor is yours.
สวัสดีครับ Hi everyone. My name is Anupha Prachumwat. I'm a researcher at Biotech, NASA, Thailand, and Centric Shrimp, Mahidol University. I would like to thank the analyzing committee of DAA11 for allowing me to share our work with you today. I would like to thank this funding agency and also organization for allowing us to be able to do our work. And also I would like to acknowledge our team, Professor Tim Frekel, Dr. Kalayasi Tunyaraksana, Professor Grant Stanford, David Bass, and other key members of our team that have been doing this work, and the staff of the two water system agricultural experimentation project of Chai Patana Foundation for allowing us to collecting data from their ponds. And the topics that I will cover today is on the shrimp culture, the whole shrimp culture pond, but at the end with emergency harvest of YHV, and I will touch on the microbiome of water and shrimp intestines. And microbiome of shrimp aquaculture recently have been got a lot of attention, especially the shrimp gut microbiome that will help to promoting the health of the shrimp. Microbiome in shrimp aquaculture here is on microbial communities in shrimp and rearing environment, and these not including just bacteria that been got a lot of attention, but also eukaryote and other viruses that been in the shrimp aquaculture system. Okay, been found that outer gut bacterial community had been observed in the diseased shrimps, and these have been shifted from the healthy to the pathobiome. But recently, in terms of the bacterial community in the shrimp aquaculture, have been limited. So the case study of the whole shrimp aquaculture production cycle have been limited. And our team have been working in one of the farm of the Chai Patana. We have been doing collaborating project with them, and we went to the collecting the shrimp sample for all various activities. That it's great opportunities for us to ask the question that we are interested in. And the question that I would like to share with you today is on how bacterial profiles of shrimp intestine or shrimp gut and pond rearing water change from the stocking of the shrimp to the emergency is explained in which YHV. And we'd like to ask whether there are any outer bacterial species, outer in terms of composition, has been used as potential early warning taxa of the YHV. Is that possible? In this pond, so the farmer usually in stocking shrimp, and then they'll culture at least 3 months or 12 weeks. But actually in this pond have been typhoon and they have the power outage, and then it's called the shrimp state, and the farmer of the Chai Patana observing the shrimp death mortality on that evening. And so the next 2 days after the power shortage and that they decided to have emergency harvest of their shrimp, which is only 8 weeks of our culture. Okay, and throughout these 8 weeks, we have been collecting sample weekly basis, and we detecting all these pathogen listed here on the left-hand side, which it happens white spot syndrome, EHP, and YHP. And this circle here that corresponding to each week, it means that we detect using the PCR whether they have this pathogen or not. This open circle is no pathogen detected, but if it few circle here that you can see on the YHP line here, we found that there been detecting of YHP in the rearing water from week 7, but not detected in the shrimp. But the sample on week 8, we were detecting both in shrimp and in the rearing water. So based on YHP have already present in the pond environment, especially in the water. And we collecting sample here that I will talk about is on water and the shrimp intestine. And here I will show you some of the part of our data that we collecting this DNA sample and send it for Illumina MySeq high throughput using V3 and V4 16S RNA amplicon sequencing. And this we put into the microbiome analysis for our question. And this is a pipeline that we did with the sequence. And here I would like to show you the principal component analysis plot of the bacterial profile of the water and then the shrimp intestine. Here that we found that there's a shift from the stocking to the emergency harvest week of both water shrimp intestine. Okay, I will show you in here that from week 0 here, the bacterial profile have been of the 2 set of sample, and it's week 3 and week 4, the bacterial profile have been chipped up to this position. So it means that the bacterial profile have been changed from each week to each week. And then week 4, you can see that the shrimp bacterial profile been shifting here through here as well, and along with the water profiles. And week 5, week 6, week 7, and week 8. See that the bacterial profile of the intestine of the shrimp have been dispersed more, or in terms of heterogeneity of the sample have been greatly more from week 6 onward. Each week, the water sample have been clustered together nicely. Okay. And then from this data that I show you, it shows that our shrimp and water bacterial profile have been different between one another. So, and the changes among weeks here, bacterial profile been larger trips, been found in early weeks, especially from week 0 to week 7. But on week 7 and week 8, where the emergency harvest occur, these have been reduced later of the rate of chip. And also the bacterial shrimp intestines, you can see that from week 6 to week 8, the bacterial profile being more heterogeneous, and especially some of the week 8 shrimp intestine sample have been much closer to the water sample in our collected data. Yeah. And in terms of alpha diversity, both water and shrimp intestine have been increased from the stocking to the culture week as the shrimp culture age. Okay, have been similar throughout the week, but actually from week 6, 7, and 8, there are more variation of the alpha diversity among the sample, especially in the shrimp intestine, that especially in week 8, have been more variation, the wider on week 8. Situation of YHP outbreak in the pond is a power outage. They've been causing these, probably reducing oxygen, have been causing the shrimp stress, but actually the YHP have been arrival on the pond environment much before the week 8 of culture. This is probably from the external source. Okay, and the typhoon that hit it into that area at that day caused the power outage and been causing stressful condition for the shrimps. And the YHP does cause why the shrimp to be weakened, and YHP been entered to the stress shrimp and replicate more. And this has been causing shrimp mortality have been observed just right before the shrimp have been on week 8. So we wanted to ask whether since the YHP have been in the pond as early as week 7, and want to check whether some of these bacterial ASV or taxon can be associated with the YHP arrival and abundance, and potentially we can use this as early warning taxa of YHP outbreak or not. So we looking at the abundance of the ASV that changed from week 6 to week 8, and we use the statistical test and supervised machine learning approach to identify these ASV. And we found that these around 11 groups of the bacterial sequences or ASV taxa that we have that might be associated with the arrival of the YHP, especially the group on this group that have been present from the week 6 and week 7 onward, and then the number been increased in the week 8. Okay, and now we go further in terms of designing the PCR primer, be able to detect this taxa in our sample in other shrimp ponds to test whether this will be able to use as early warning signal for the YHP. And I'd like to give you the take-home message that the pond microbiome, especially in the rearing water and shrimp intestine, have been shifted from the stocking to the harvest day of the shrimp. And different bacterial profile been observed between rearing water and the shrimp guts, between in the shrimp microbial community. And the bacterial profile been tripped at a larger rate in the early week when the in accordance with the biomass has been increased in the pond. And shrimp intestinal microbiome become increased heterogeneity from week 6 onward, and to the when power outage promoted shrimp mortality, the shrimp been stressed and YHP have been entered into the shrimps because the YHP have been detected that are already present in the pond. And I also showing you that there's some bacterial ASV that potentially as early warning of the YHP outbreak in the pond. That might have to be studied further. Thank you very much, and I'd like to take questions that you might have.
Thank you, Dr. Anupap. Can you tell us with your emerging signal you're talking about, how many taxa as a candidate for this result?
Currently, we have about 11 taxa, but including several ASV in each taxa, and most of these groups are uncultured bacteria. Yes.
Okay. Dr. Suplani asked where YHV come from on week 7.
I believe, but actually we don't know. We have to look into further. But with the sample that we have collected, but so far we look at through the week from to week 7 and we detect in the water. I suspect that it might come from agitation of bird or other external source that might have the YHV already present in the pond, because it's it's not a closed system, so it's open pond, outdoor pond. So that might be the cause. Or another possibility is that YHV is present in the pond for quite a long time and then have very low prevalence. But when the shrimp, what I believe that when the shrimp have stress and because of the lost power and then low OD, that might cause the shrimp to die. That might be the case.
Okay. Is it a biosecurity system?
At Chai Patana, the staff tried to, but since it's opening outdoor pond, so very hard to keep those external salt, especially from the frying stuff. And only the lining of the wall of the pond that was lined with the PE, but the bottom is still open, like the earthen pond. So that would answer your questions. I hope that answer. So, okay.
Thank you very much, Dr. Anupap. We
Thank you
for this emerging signal.
Thank you very much. Happy to be here.
Bye bye.